Hi guys, welcome to Science Chomp. Today we're going to be talking about transcription and translation. Now bear in mind this is for grade 8, but if you're doing something like A-level, maybe this is also going to be kind of useful to get the basics. Okay, so first of all, let's have a think about what transcription and translation actually is. So as you can see on the page, transcription is transcribing the DNA to make an mRNA template. What that means is that if you imagine DNA like a book, mRNA template would be like a photocopy of one page. So it's like you're transcribing one piece of DNA. Now, that's transcription. Translation is converting that mRNA into a protein. If you have a look down here, we have our cell. DNA lives inside the nucleus basically, and this is where transcription happens. And then once the piece of mRNA is made, it gets modified and it leaves the nucleus and it finds itself one of these ribosomes. And ribosomes are basically where the translation takes place, and you end up with this long, what we call polypeptide, which is an amino acid chain. Ribosomes can also be found in endoplasmic reticulum, or they could be free floating within the cytoplasm. Okay, so let's get into some of the details of firstly transcription. So within your DNA, so this is your DNA double helix, you got sections called promoters and terminators. What a promoter does is it causes the RNA polymerase to bind. Once it binds, it unravels the DNA. Your DNA is this black line here, and it starts to add bases from the 5' to the 3' direction. It's basically adding the thing like this. So it's starting from this end and it's going in that direction, adding the bases like that. Okay, as the thing moves along. And so this strand is getting longer and longer and longer, but because it's being added in that direction. That's the general idea, okay? Okay, so our DNA template is the blue one. And you have to bear in mind, with DNA, when you got the double helix, you got these complementary bases. So those complementary bases are down here. So whenever you have an A on the other strand, you have a T, and vice versa. Same with C and G. So G is always with C, A is always with T. Now, when you have RNA, we don't have this T, this thymine doesn't exist. Instead, we have a base called uracil. So whenever you have an A, you're going to have a U up there. Whenever you have a T, you have an A. Whenever you have an A, normally for DNA you'd have a T, but here you'd have a U. And we can just fill in the rest of them like so. G, G, this one's C. A is going to be a U, another U over here. C is going to be with G. G is going to be with C. T is going to be with A. A is going to be with U. U again, and A. What's it? C is a G, G, and then an A. So this is your complementary mRNA strand. That's your complementary mRNA strand. But what do all of these bases do? Well, these bases, they code for amino acids. Every 3 we call a codon. So let me just get another color over here. So every 3, you got 3 over here, that would be a codon, and a codon codes for an amino acid. Once you've made your mRNA, you got actually sections of the mRNA which don't really code for anything. They have to be removed. They're called introns. And you got to put caps on either end of the mRNA as well. So this is called processing. The mRNA needs to be processed. And once it's processed, what happens is it leaves the nucleus, just flies out, and it finds one of these things, a ribosome. On this page, we have a whole load of codons. This is what we call the codon table. And this is your mRNA strand from earlier. Let's figure out what all of these actually code for. So remember, these are every 3 we call a codon, and this is the codon chart. So the first one is AUG, and you can see it says MET. Now this is quite convenient because MET is the start codon. So this gives the signal to the tRNA. So the tRNA is going to come along and it's going to see that, and it's going to connect on, and it's going to start the entire chain. And the tRNA, as if you remember from before, kind of looks a little bit like this. It's got these like round things like so. You got an amino acid on the end. I'm going to write AA for amino acid, right? And you have on this end over here, you're going to have an anticodon, which is going to be like complementary to this. So if you got AUG, on this one, you're going to have UAC. Okay, that's going to be the anticodon for this bit. It's going to connect on. It's going to provide the amino acid. In this case, MET, that's the start one. Okay, so let's figure out what GCU is. We have to find GCU, and it's glycine, I think. UGC, where's UGC? It's CYS, cysteine. AAU, where's AAU? AAU is ASN. I forget what that stands for. And then UAG is your stop codon. So this is where it stops. So you make this like small amino acid chain, and that's where it stops. So it starts there and it stops there. Okay, well, what does that actually look like? Remember, this is going on in the ribosome, and you have this thing happening. So you got your ribosome, you got 2 units. You got a small unit here and a big unit up top. And what's happening is rRNA is what the ribosome is made of. And you have your tRNA, which are these things. And remember, your tRNA looks like that. It comes and it joins and it supplies an amino acid. And once it connects, another one connects on. And what you end up with is this chain being connected onto that one. And then the next one comes along, and then it connects onto that, and you end up with this long chain of amino acids. Once it hits that stop codon, what happens is the whole thing dissociates. Okay, it basically, you end up with just this chain. It basically comes off of the mRNA. So you end up with that chain, and then it folds itself into the shape of whatever protein, and it's connected with like the shape is held with bonds like that. And yeah, that's how you make a protein. So this bit is called translation. So remember, with translation, you got your mRNA over here. This is your template. You got your tRNA, which is this thing over here. tRNA looks like this. This is your tRNA. It stands for transfer RNA. Your ribosomal RNA is made up, what makes up the ribosome itself, and it allows the growing chain to basically form. And once that's formed and it hits that stop codon, it disassociates. You have that long chain of amino acids like so, which then folds itself into a protein. And that's it. It's as simple as that. Thank you very much for watching, and goodbye.